Hi friends, welcome to Field Crop video series. In this video, I'll talk about cowpea. The scientific name of cowpea is Vigna unguiculata. The chromosome number is 22, which appears in diploid form. It is called as black-eyed pea, field pea, southern pea. Earlier, the English name was cavalens, which was popularly called in West Indies. Word lobia derived from Greek lodos means pods. It is a day neutral crop. In India, it is called in Bengali as barboti kolai, in Kannada alsande, in Hindi lobia, and like that in different colors, in different languages it is called with different names. Most important food legume. It fixes atmospheric nitrogen through its root nodules. It is mostly grown in semi-arid tropics covering Asia, Africa, Southern Europe and Central and Southern America. It is compatible as an intercrop annual legume. Plant types are often categorized as erect, semi-erect, prostrate or climbing. It is grown on wide range of soils. Cowpea is considered more tolerant to drought. The history of cowpea dates to ancient West African cereal forming 5 to 6,000 years ago. Importance of cowpea. It has multiple purpose used as a vegetable. Cowpea seed is a nutritious component in human diet, livestock feed, etc. It is also a popular green manuring crop, also used as fodder purpose as a cover crop. In Africa, the young leaflets are eaten as spinach and dried leaflets are used in soup preparation. Haulms are fed to livestock. Coming to nutrient composition, it consists of protein of 24.8%, 63.6% carbohydrates, 1.9% fat, 6.3% fiber, 3.8% ash, 0.00042%, that is in minute amount of riboflavin and niacin. Taxonomical classification. Cowpea belongs to kingdom Plantae, class Dicotyledon, subclass Polypetalus, order Rosales, family Fabaceae, subfamily Papilionaceae, genus Vigna and species Unguiculata. The different species description. Cultivated genus Vigna is classified into 4 subspecies. Those are Vigna unguiculata subspecies cylindrica, that is catzung bean. It has most diverse and widest in distribution. Second is Vigna unguiculata subspecies Decintiniana. Third is Vigna unguiculata subspecies Secupendalis, that is yard-long bean. The last one is Vigna unguiculata subspecies unguiculata, that is black-eyed pea, most popularly grown across the country. Wild species of Vigna are Vigna unguiculata subspecies Decintiniana and Vigna unguiculata subspecies Menesenes. Cultivated species are Vigna unguiculata subspecies Sesquipedalis, Vigna unguiculata subspecies cylindrica and Vigna unguiculata subspecies unguiculata. Botanical origin. Wordcourt recognized the progenitor of cowpea during 1970. The present cultivated species origin from wild species, that is Vigna unguiculata subspecies Decintiniana. Origin of cowpea is primary center of origin is India, secondary center is Africa and China. Places of domestication are Ethiopia, Central and South America and West Africa. Coming to botanical description. Habit is annual, twinned or twinning, erect to climbing to about 3 feet, determinate, highly branching. Roots are strong taproot with numerous lateral roots. Stem is cylindrical, slightly ridged, almost glabrous and somewhat hollow. Leaves are alternate, petiolate, trifoliate. Leaflets are ovate to entire, short hairs on both the surfaces. Inflorescences are axillary, raceme with flowers, congested at the top of the nodes with 2 to 5 flowers. Flowers are showy, white or yellow or pink or light purple in color. These are bisexual, hypogynous, zygomorphic, complete, pentamerous. Sepals are 5 in number, gamosepalous. Petals are also 5 in number, polypetalous. Corolla is papilionaceous. It has 3 different kind of petals like standard, wing and keel. 10 stamens, diadelphous, that is 9 plus 1 arrangement. 9 are free and 1 is fused. Then filaments alternatively long and short. Anthers uniform, bright yellow in color. Stigma are captate type and style is terminal. Ovary is superior, unilocular with many ovules on marginal presentation. Pods are smooth, 6 to 10 in long, cylindrical and generally somewhat curved. Pod color may be distinctive, most commonly green, yellow or purple. Seeds are light, 8 to 20, vary in size, maybe 2 to 12 mm, shape globular to kidney shaped, then texture smooth to wrinkled, and color they are loosely packed. Different colors and loosely packed. This is a flower and different labeled parts. Floral diagram. It consists hermaphrodite flower, calyx 5, corolla 1 plus 2 plus 2, androecium 9 plus 1, superior ovary with 1 gynoecium. Anthysis. There are 2 to 3 flowering periods. It takes 11 to 14 days for the flowers to develop and bloom. Flower opening at morning from 6 to 10 am. Dehiscence of anther period to blooming, that is 10 pm to 1 am. Coming to breeding objectives of cowpea, first and primemost is yield. High green pod yield, then high seed yield, fodder yield, dual purpose. Dual purpose is both for consumption and fodder, then appropriate plant type, resistant to disease and resistant to insects. In nutshell, we can say resistant to biotic and abiotic stress. Breeding methods. First is introduction. Method is introduction. Through that, 2 varieties were developed, that is Iran cowpea and Russian cowpea. Through selection, PLS of CO1 from C57. Through hybridization, intervarietal hybrid CO6, CO2, then interspecific hybrids were developed between Vigna unguiculata into Vigna vexillata. Through mutation breeding, CO5, IT84, E124. Coming to hybridization methods, first one is selfing. Cowpea is being a self-pollinated crop because of cleistogamy. It does not require any artificial selfing methods. To prevent the outcrossing, we generally go for bagging of mature flower bud. Crossing technique. Cowpea flowers are large and showy. Emasculation carried out in mature flower bud in preceding evening. Then pollination is done simultaneously or in the next day morning from a freshly opened flowers. Emasculation. For this, select young buds in an inflorescence and remove all immature buds and split open the keel petals and remove the stamens one by one, holding the filaments. Cut about 2/3 the worth of the unopened bud in the center. The style and stamens, which must not be injured. Remove 10 anthers and cover it with butter paper. This is how the emasculation has to be done, by carefully removing the petals and the anthers. Coming to pollination, pollination is done in the next morning from protected fleshy opened flowers. The standard and wing petals are removed by slightly depressing the keel. The stigma covered fully with large quantities of pollen will protrude outside. This can be used as a brush for transfer of pollen to the emasculated flower. Like this, pollination has to be done. The powdery like pollen taken with the help of a painting brush, then dusting it on the stigmatic portion of the female selected flower. After that, better to cover it with a cotton plug in order to avoid the cross contamination and enable better pollination. Released varieties. For vegetable type varieties, Pusa falguni, Pusa barsati, Pusa dufatak. For fodder type, Russian Giant T2, K397, Chimera, HFC 42-1. Varieties for different states are given in this list. The popular cowpea varieties are Pusa basant, Pusa falguni, Arka garima, Arka sunaman, etc., Varun and all those things. Disease resistance. The major diseases are caused by Cercospora leaf spot. For that, resistant varieties are AK 963-1. Then for fusarium wilt, Victor, Brafum. Then for charcoal rot, Vector and iron. For powdery mildew, Chinito. And for rust, Pusa barsati. Viral disease and its resistance. Cowpea mosaic is a major disease. For that, the resistant varieties are TVU 39, 45, 99, then TVU 612 and all those things. For pest and disease resistance, for leaf hopper, TVU 59, TVU 190, for aphid, TVU 140, TVU 2740, and for thrips, TVU 1509, for pod borer, TVU 946, pod sucking bug, TVU 1977. Major pests are root knot nematode, aphid, green stink bug and pod borer. Major diseases are leaf spot, rust, southern blight and fusarium wilt. Germplasm conservation. More than 5,000 germplasm lines are being maintained at International Institute of Tropical Agriculture IITA, Ibadan, Nigeria. At International Cowpea Disease Nursery, national collection of cowpea lines in India are 3,000, Ghana 1,000 and Pakistan 500. In India, this germplasm conservation is done by IIVR Varanasi, Indian Grassland and Fodder Research Institute, Jhansi. This mainly work for fodder purpose cowpea.